Hello friends and my dear students. We are discussed about the topic meiosis. Meiosis, it is the reduction division, and the meiosis is very longest procedure or longest phase of the cell division. And the meiosis takes place in the reproductive cell, or we can say that the germ cell are divided and form a new cells by the process of meiosis. Meiosis has two subtypes, meiosis I and the meiosis II. The meiosis I is very longest process. There is the cytokinesis I as well as karyokinesis I. So in the meiosis I, there is karyokinesis I divided into four phases: prophase I, metaphase I, anaphase I and telophase I. And the telophase I is followed by the cytokinesis. And in the previous lecture or in the previous video, we discussed about the meiosis I, and at the end of meiosis I, there is the formation of two cells. There is the formation of two cells, and the meiosis I is heterotypic division. It is heterotypic division because the two cells which are formed at the end of meiosis I, these two cells are different from their parent cell. These two cells are different by their parent cell, as well as the chromosome numbers is also half to their parent cell, and these two cells are undergo the second phase or the second stage, that is the meiosis II. At the end of meiosis I, we get the two cells with non-sister chromatids because the non-sister chromosome, because the chromatids of each chromosome is exchange their segment. The chromatids of the chromosome is exchange their segment, and the formation of two daughter chromosomes, that two daughter chromosomes, and the formation of two cells, finally the formation of two daughter cells. These daughter cells now undergoes the process of meiosis II, and the meiosis II is the homotypic type of the division. Why it is known as the homotypic type of the division? Because it is equal to the mitosis. It is equal to the mitosis, means from those two cells, formation of the total 4 cells, and this formation of total 4 cells means the 2 from the each respectively, and those cells are similar to their parent cell. That is why it is known as the homotypic type of the division, and the meiosis I is the heterotypic type of the division. Now these two cells are undergo the next process, meiosis II, and in between the meiosis I and meiosis II, there is very smallest resting phase. In between the meiosis I and meiosis II, there is the smallest phase which is known as the interkinesis. That interkinesis is the resting phase The interkinesis is present in between the meiosis I and meiosis II. It is the resting phase of the cells, and after the interkinesis, the meiosis II is occur, and in the meiosis II, there is again divided into karyokinesis and karyokinesis and the cytokinesis. Again, the karyokinesis in the meiosis II is divided into 4 phases, that are prophase II, metaphase II, anaphase II and the telophase II. Okay, so the two cells after the meiosis I is undergo now the process of the prophase II. In between prophase II and the last meiosis I, shortly the interkinesis, and these cells are undergo the prophase II, in which in the prophase II the daughter chromosomes is present, or the chromosomes with exchange segment. Okay, the chromatids of that chromosomes is exchange segment, and it is distinct, and the centrioles try to away to each other, and these centrioles are away from each other and they are arranged at the pole region. The centrioles are arranged at the pole region, in which the nuclear membrane and the nucleolus, which are reappear in the telophase I, these are the disappear in prophase II. The nucleolus as well as the nuclear membrane both are also disappear in the end of the prophase II, and that prophase II is undergo again the next division, that is the metaphase II. In the metaphase II, the formation of spindle fiber, or we can say that the formation of kinetic fiber is carried out, and the centrioles are arranged and it present at the pole region. The both centrioles are completely away from each other and they are arranged at the pole region, and in between the two centrioles there is the formation of spindle fiber, and on that spindle fiber the chromosome are arranged. On the spindle fiber or on the kinetic fiber, the chromosomes are arranged and they are present in the equator. They are present in the equator, and the both chromosomes are present in the middle. We know that in the metaphase stage, the formation of metaphase plate, and on that metaphase plate the chromosomes are arranged. These are present at the equator or the equatorial region, and in the middle of that cell or in the middle of that what this kinetic fiber. Okay, this is the metaphase 2. After the metaphase 2, there is the next process in which the cells are undergoes the third phase, that is the Anaphase II. Anaphase II. In the Anaphase II, the centrioles are away from each other, they are arranged and they are present at the pole region, and the chromosomes which are arranged at the equator or which are present in the middle of in the metaphase 2, the centromere of the chromosomes are divided into two. The centromere of that chromosomes are divided into two, and the chromatids are again divided into haploid, means from the total 4 chromatids, it divides into 2 with each centromere, and here the daughter chromosomes are formed. The daughter chromosomes are formed, which is one is the sister chromatids and another is the non-sister chromatids, means it is with the exchange segment. It is with the exchange segment. Here the daughter chromosomes are formed. At the anaphase stage, we know that the daughter chromosomes are formed and they are pulled to away each other. They are pulled away to each other and they are arranged at the opposite pole. The both daughter chromosomes are away from each other because they are pulled to each other, and they are arranged by the kinetic fiber at the pole region. The both daughter chromatids are arranged at the pole region with the help of that kinetic fiber, and that fiber is known as the interchromosomal fiber or the chromosomal fiber. The anaphase II, there is the 4 daughter chromosomes are formed. They are undergo the process of telophase II, and at the telophase II stage, the formation of 4 daughter chromosomes, and these daughter chromosomes are arranged in the 4 nucleus, means the chromosomes are uncoiled by the nuclear membrane, and there is also reappear of nuclear membrane. Along with the nuclear membrane, there is the nucleolus. Both are present and both are the reappear at this stage of telophase II. And telophase II, it again undergoes the cytokinesis II, and at the end of telophase II, cytokinesis II, total 4 daughter cells are formed. At the end of meiosis II or at the end of telophase II and cytokinesis II, there are the total 4 cells are formed with nuclear membrane, nucleolus, as well as the daughter chromosomes. The daughter chromosomes are different in the each cells. Here the exchange with the daughter chromosome with exchange segment of the chromatids, and the chromosomes are as it is chromatids, and the end of telophase II and the cytokinesis II, we get total 4 daughter cells, and these 4 daughter cells are formed at the end of complete process of the meiosis. Means the single cell are undergo the meiosis I and meiosis II. At the end of meiosis I, we found the 2 cells are formed, and at the end of meiosis II, means the complete division or the complete meiosis II, lastly we got what? We get the total 4 cells. From the single parent cell, formation of total 4 haploid cells. 4 haploid cells with exchange segment and with different chromosomes, and it gives the variations. The meiosis process is occur in the reproductive cell, or we can say that in the germ cell. The germ cell or reproductive cell, we know that the sex cells, these are divided by the meiotically, and lastly it forms the 4 cells from the single cell. And if it is absent, the meiosis process or the meiotic division is absent, the chromosomes or the cells are abnormal, because here is the haploid chromosomes from the diploid chromosomes, or the haploid cells are formed from the diploid cells. If it is absent or the meiotic division is absent, then maybe the cells with quadruple chromosomes or quadruple or the double, and it is the abnormal type of the cell. That's why the species number constant or the constant number of the chromosomes is given in the particular species by the process of meiosis. And in the meiosis process, the genetic material or the exchange of the genetic material takes place, and it is the raw material for the evolution. That's why the genetic material is exchanged, that's why the variations are occur, and these variations which present in the genetics or in the genetic variation occur, this is the raw material for the evolution, and it is the biggest role in the evolution. Directly we can say that the meiosis process is the basic need for the genetic variation, and here the complete cell division is over. The mitosis, meiosis I and the meiosis II. Thank you.